नमस्ते, हमेशा खुश रहो बिटिया. I am JVN Dr.LILY Trivedi, and today I am going to give a lecture to you on anomalous secondary growth. Now this is a lecture for BSc.B.Ed ZBC 4th semester, and the course is Anatomy, Ecology and Evolution. So the session is anomalous secondary growth. Now we all know secondary growth, but now we are going to study what is anomalous secondary growth. As we all know, in monocotyledons, the secondary growth is absent as the vascular bundles are of closed type. That means the cambium is absent. But in some plants like Dracaena, Yucca, Aloe arborensis, Agave, etc., exhibits secondary growth, which is anomalous in origin as well as in function. Now, how can there be secondary growth? There are certain conditions. So, what are those conditions? Anomalous position of vascular cambium. Unusual shapes in stems are formed by the unusual position of cambium. So, such examples are seen in Bohnia, Langstoffiania, Turgenia, and Thinonia. Second is abnormal behavior of the normal cambium. Now, certain segments of cambia cease to produce secondary xylem, but at the same time, they produce secondary phloem on the outer side. So, what that happens? There is no normal activity, so there is ridges and furrowed staves are formed. So, the example for this is bignonia. Third is successive cambium. In plants like Cycas, Genitum, Avicennia, a number of cambial rings arise in succession and they produce concentric rings of secondary tissues. Now we have another type that is interxylary or included phloem. Here the secondary phloem is found in the form of strands or islands and it is embedded in the secondary xylem. So therefore it is called included phloem or interxylary phloem. The example is Salvadora, Combritum and Strynicos. The fifth type is presence of medullary bundles along with normal cambial activity. Now here what happens, the normal vascular bundles are present in a form of ring and the additional vascular bundles are found scattered in the pith region or medullary region. So that is why they are called medullary vascular bundles. An example is Boravia. The sixth type is presence of cortical bundles along with normal cambial activity. So in Nyctanthus, what happens? A ring of vascular bundles from which a normal cambial ring is formed. But in addition to this, vascular bundles in the cortical region also vascular bundles are found. Then we have intraxillary phloem or internal phloem. Usually what happens, primary phloem is formed outside the cambium. But in some plants, primary phloem is on the inner side along with the xylem towards the pith. So such a phloem is called intraxillary or internal phloem. So example is Solanum tuberosum. Then we have secondary growth in monocots. So as discussed, in some monocots, the stem exhibits secondary growth. So in Dracaena is a classical example. What happens here, there is secondary thickening meristems originate, where from the ground tissue outside vascular bundle. Now these meristem cells cut off cells inside from additional vascular bundles and are formed along with parenchyma cells where in between the vascular bundle. So they consist of only one type of cells, unlike the normal vascular cambium. But these secondary thickening meristems do not produce continuous diasm on inside, but only on the vascular bundle with xylem and phloem. So no phloem is formed on the outer side. So there are two plants, Dracaena and Boravia, which we are going to study in the next coming lectures. Then the next is, ninth is anomalous primary growth. That means absence of vessels in the xylem. Usually what happens, vessels are found in xylem of angiosperms. But in some plants, vessels are lacking. For example, Hydrilla and Binteraceae family. Scattered vascular bundles along with cambial activity in dicot. In dicot stems, what happens? Normal vascular bundles are arranged in ring, but some dicot stems also possess scattered vascular bundles in the cortex like monocot stem. So such vascular bundles are found in Piper and Peperomia. So I have taken my matter from these references, and I hope you have liked my lecture. So I conclude with this, and in the next lecture we are going to study the detail of Boravia and Dracaena, anomalous secondary growth. So students, if you have any query, any comments, please do comment in the comment section box. I will resolve it soon. This session was powered by 2.0 version of Jayoti Vidyapeeth Women's University. Thank you. Namaskar.